It's my pleasure to introduce Dr. Billie Swalla, who needs no introduction, from the University of Washington. Billie's Twitter handle is Evo Devo Diva. I'll tell you how she got that title. Back in the 1990s, a group of us at the ASZ, SICB, were trying to figure out how to start a new division of evolutionary developmental biology. Do we separate, and have a separate thing, or do we have it from the existing developmental and cell group? What are we going to do? And we had to find a way to get together and talk about this. And so a group of us got together at 7:30 in the morning, because that was the only time we could get together. And those of you who know Billie know she's not great at 7:30 in the morning. We were sitting around a table waiting for breakfast, and Billie wants coffee. No, she doesn't want coffee. She needs coffee. And she's trying to get the waiter's attention. Waiter, I want coffee. Finally, after a few shouts, the waiter turns, comes over to our table, takes the silver carafe, and pours coffee for her. And so it was noted, the queen has been served. And from there, we did form such a group. Billie became the first program director of the Division of Evolutionary Developmental Biology. She then became the president of the Society for Integrative and Comparative Biology. She became the first woman director of the Friday Harbor Marine Biology Lab. She has pioneered the inclusion and outreach programs in this area, and she's also, of course, advanced our knowledge enormously of the non-vertebrated deuterostomes, especially the tunicates, where both the heads and the tails of the tunicates have been thoroughly analyzed. And she is really so well known for this incredible group of organisms that she studied, and which are the ones by which Kovalevsky really introduced this notion of evolution and development and connected the animal kingdom, the vertebrates and the invertebrates, through the larvae of tunicates and Amphioxus. So we are going to give the Service Award. Now, Billie, when she gave the first Service Award, said that she really wished that it had a better name. Service, kind of is quotidian, perhaps even menial, that it should have something better, and I agree. I think that maybe the Pan American Society for Evolutionary Developmental Biology might want to call this the Notochord Meristem Award. It's the award for people who do the things that allow other people to do more things, and who really make the conditions possible and induce the conditions such that the affairs of the society can continue. If the society is like an organism, Billie Swalla has been the notochord of the society. So Billie, come on up.
All right, well, I used to joke, somewhat joking, that I had to create an entire society to be able to do the kind of work that I wanted, but in a way, it's really no joke. When I was a graduate student, nobody was thinking of evolution and development. I was a cell and molecular biology trainee and also went to the journal clubs of the Ecology and Evolution group, and people didn't really understand that. They were like, what is she doing? Like, what are you talking about? And that was before we knew what genes were involved in development. And I was just sure, like many people were sure, that once we got those genes, we were gonna find out that there were some underlying things for body plans, and that we were just gonna continue to go from there. So in my lab now, I work on two different levels. One is at the, whoops, I'm gonna see, this is gonna go fast. One is at the microevolutionary level, where we work with very closely related species, and the other one is at the macroevolutionary scale, which I'm really interested in, the evolution of chordates. And I started out working on ascidians because I believed that they would tell us about the evolution of chordates. And since my lab also works on hemichordates because I think they're a better model to tell us about the evolution of chordates. And so in 2000, really in the mid-1990s, I started to do phylogenies because the groups I work on, the tunicates and the hemichordates, have almost no fossil record for the solitary hemichordates and the tunicates. And so we started doing molecular phylogenies, and looking at these molecular phylogenies, I went to give a talk, and they told me, tell me what you think was going on, and I said, I think people are wrong that there's a tunicate ancestor there at the base of the deuterostomes. I think it's something worm-like. I think it would be something worm-like, like hemichordate-like, and the other groups are worm-like, so why not? For the next few years, every time I gave the talk, people just said, that can't be, like, how dare you? People give an entire courses on how you get a chordate from a stocked animal, and no one had talked about a worm-like ancestor. But I would say now that most of our data suggests all of these different, we have groups in each of these that are sequenced from the genomes, and I would say that most people agree that there probably was a worm-like ancestor, the deuterostomes. I'm not going to talk more about that today. I'm going to talk about the microevolutionary stuff that we do. And just to say that one of the ideas was Garstangs, that you had echinoderm larvae that made a hemichordate larvae. That's pretty easy to do, but this was always really the hard step right there. Kind of go from an larvae to an ascidian larvae. And the truth was, you'd really want to do that because the larvae are really adorable. And then Romer was in most of the textbooks. I hope it's out of the textbooks now. I've been working on doing that. But he started with a colonial hemichordate here, and then it was really hard to make a solitary hemichordate. Pretty easy to make an ascidian from that, but a little bit harder to make the vertebrates from that. And so what we said in 2000 was that there was an ancestor that was worm-like, and that you had these very different changes to the echinoderms and chordates on the other side. And if you start with a worm-like ancestor like a hemichordate, it's really not that hard to make a chordate. So we're still trying to figure out exactly how all that goes. So I started, again, interested in chordates. I saw this picture of conklins in a textbook. It's a true story. And I was in developmental biology, loved developmental biology. And I said, that's the secret of life, you know. These are Styla partita embryos. They're embryos that have colored cytoplasm. A few species of ascidians have these colored cytoplasms. I like, this is going to tell me the secret of life. That's what I've been studying since. I haven't found out the secret of life, unfortunately, but I'm having a good time looking for it. So I wanted to just call out some of my advisors here. Mike Solers was my graduate advisor. He had a very unusual lab in that there was both chick embryos and sea urchin embryos. And he would never let us just say in chick this and sea urchin that. He'd be like, no, talk about cell movements, talk about mechanisms. Don't be talking about the specific thing you work on, talk about developmental mechanisms. He also said some good things, keep the microscopes covered. My students will tell you I still complain about keeping the microscopes covered all the time. Eat chocolate every day. I still really love chocolate. Don't lose your career when you have a child. He and I had children at the same time, and it was a good thing for me because as a graduate student, and your advisor has a kid the same age, we could talk about them getting sick, et cetera. And he also said everyone should work on Christmas because he was Jewish. So I always say this, that you know, take some advice from your advisor, but you don't have to take all the advice. You can, I would say, Michael, it's a holiday for most of the rest of the world, you know? I'm gonna take it off. Because I saw that picture in the textbook, I told my family I have to go to the embryology course, and I went to the embryology course in 1983. It was amazing, changed my life like it has many people's lives. The course instructors were Bruce Brandhorst and Bill Jeffrey. And then I ended up getting my PhD with Michael Solers. I was working on chicks at the time. And From there on, went out and I was like, okay, now I'm going to do this evolution development that I've been wanting to do. And to me, they always made a lot of sense together. There was a ASD symposium that I organized in 1992, and I felt like no one was going to ask me to be in a symposium, so I just started organizing myself. I still like to do that, so you can see that. In 1994, there was a symposium on origins and evolution of body plans at the University of Indiana, organized by Rudy Raff. And we decided to get an email list to disseminate information and coordinate activity. And so Rudy Raff, Greg Gray, and Jim Henkin decided they were going to get together and do this. The list was at the University of Colorado. That list still exists. People still post there. I can't post there anymore because they changed my email address at University of Washington. And we're starting a new one at the University of Washington, and you'll hear about that soon. So I feel like since I got this award, I better do something to deserve it. So that's what I'm going to start doing. The main thing that we did in 2000 was start the SICB Division of Evolutionary and Developmental Biology. Scott remembers that. Our planning for that, we had 2 symposia, the Hawk Symposium, History of Evolution and Development. It's really a great way to start out the division. It was in 2000. I was like, this is great, turn of the century, so we can remember the date. And I also sang my talk that year because Scott had written down this 50 ways to lose your larva. And so I sang the talk because I wanted something to happen there. And I forgot I didn't have tenure, and somebody from UW, I just moved to UW, said, oh my God, you sang your talk. And I thought, oh my God, that's it, my career's over. But fortunately, they had a sense of humor, so it all worked out. And the people who saw it, and I never did it again, so that was that. In 2005, the Europeans started their Evo-Devo list. Whenever you start something, I found this out when we started the SICB division, a lot of Europeans were like, why are you leaving us out? And I said, we're not leaving you out, start your own. And so they started their own, and we're talking about now how we have, we're trying to have the meetings every other year so that we can all go to all of them. And then in 2013, there was an SF workshop to begin the Pan Am Society for Developmental Biology, or Evolutionary Developmental Biology. And this was the first meeting. It was at Berkeley, California, and Nipam Patel, Chris Lowe, Karen Sears, and Ehab Abouf were organized that. Ehab was our very first president. Karen was the second president. I was the third president. Chris was fourth president. And so it's been a really fantastic time, and we really are really thankful to SDB for meeting with us as well. I think it's been a great meeting, and really excited about it. So now I want to tell you a little bit about things that I do. This is the microevolution thing that we do. So you are all chordates. You all have an anterior head, a posterior muscular postanal tail, notochord, and I love being called the notochord, Scott. That's a great thing for me. A hollow dorsal nerve, which allows the notochord to roll up, and then pharyngeal slits, which tunicates don't have, but they will have once they metamorphose. So we wanted to go to Roscoff, France, and look at these tailed and tailless ascidians that people had been talking about, and see what we could do there. So what was cool was these had been described by Barrel, and he talked about this tailed ascidian called Mogula oculata, a tailless ascidian called Mogula culta. You notice that there's only a few letters difference between their names, so I usually call them tailed and tailless. It really gets confusing in my lab because sometimes if you don't label the tubes just right, nobody can tell exactly what's there. But one of the amazing things was the first year we were there, like any developmental biologist, I'm like, hey, we should try crossing these. So we crossed them, and they take about 12 hours to develop, so it was late at night, everybody had gone home, and I'm looking through the microscope, and I was like, I swear I see some little eyes in those guys there. So if you take the hybrid and you take the tailed species, the top one, it always develops in a tailed tadpole way, which is very similar to what we know about ascidians because they have a lot of maternal factors that determine how they're going to develop. And then if you take the tailless one and you take the tailed sperm, what happens is you get these hybrids. The hybrids now have a little tiny tail. That tail is made up of 20 notochord cells. Trying to get this up here without going to go forward. And it also has now an otolith that's re-expressed. I'm going to show you that. We know now that those pigments are pseudogenes in the tailless species. That's why we never see anything in the tailless species. In the tailed species, that gene is intact. In the hybrids, we're seeing that tail gene that's coming through there. And what I'm going to tell you about is that there's a number of larval genes we see now that they've become pseudogenes in the tailless species. It's probably going to extinction, but we can use this system to see how genes turn on and off in these close related species and kind of see how gene networks fall apart, which is a really interesting kind of thing. So it's late in the day, and if you want to take a little nap, that's okay. I'm going to just tell you the bottom line, so if you don't want to see the data, that's okay. We found changes in gene expression in regulatory genes. We found downstream structural genes that have become pseudogenes, and there's been a number of these. The muscle-actin genes that are in the tadpole larva are pseudogenes. I told you the tyrosinase and tyrosinase-associated protein. We found some neural genes that have become pseudogenes and DDX517. We found heterochrony and changes in the timing of gene activation, especially in the metamorphosis genes. And we thought we found gene loss, but actually when we analyze it, it turned out there's a gigantic transposon that had just knocked out this maternal tyrosine kinase. And when we look for these changes, some of them we find in the other tailless species within this group of mogulas. There's lots of tailless species, and sometimes we find them and sometimes we don't. And we're still trying to look for the smoking gun for the one that we think is the right one. So this is, again, just telling you the tailed species has a head, tail, otolith, it's chordate. The hybrid has a head, a short tail, and an otolith. And the tailless ones lacks the chordate features, head, tail, and otolith. And Richard Strathman calls it unchordating, so it's losing its chordate features. This is showing you that within the Mogulidae, so there's about 3,000 species of ascidians, but it's only within this one family, the Mogulidae, showing at the bottom there, that we have these two clades, Woods Hole clade and Roscoff clade, and in those clades there's multiple species that have that are tailless, so that we can find out whether it's happening all the time. And then we have the genomes now of Occidentalis, which is kind of an outgroup, a tailed outgroup, and we can get that year-round from Florida, and then the tailed and tailless species from Roscoff, France. And then we also have transcriptomes as well. So this is the beautiful Boltinia. That's the species that we have in Friday Harbor. Again, gorgeous orange crescent. I love showing this to embryos because once it's fertilized, you can tell what's anterior, posterior, dorsal, ventral. And so they get the whole idea of how embryos develop. The bright orange there will go into the tail muscle cells. We know now that the reasons that they become tail muscle, this is something Conklin was very interested in, is because there's a T-box transcription factor that's called MACHA1. It was cloned by Hiroki Nishida in Japan, and when you have a certain nuclear cytoplasmic ratio, MACHA1 goes into the nucleus and makes them become muscle. So, and then after they swim around, then they turn into these little, oops, go backwards here. They turn into these little spaceships, sit down on the bottom of the dish, and then they'll make a little tunicate. So it's kind of late here in the day, but I thought I'd show everybody what it looks like when a tunicate develops. You can always tell a tunicate embryo, even in the plankton, because they have all these cells on the outside. The very outside cells are called the follicle cells. And then there's a chorion that's kind of bulletproof, but we can get off. And inside that's the test cells. The test cells are always really, really active. Try not to look at the test cells. I always tell students, don't look at the test cells. Look at the embryo that's on the inside there. You can see the plane of bilateral symmetry. It's kind of off to 11 o'clock or so there. And this embryo right now is gastrulating. It's gastrulating from the vegetal pole. The endodermal cells go in. And so we're looking down on what's going to be the dorsal side. Pretty soon you'll see it start to roll. They do roll. They have nodal on the left side, and they're going to roll to the side, and you'll see that the neural tube is zipping up, and it zips up from posterior to anterior rather than anterior to posterior the way it does in most of the rest of the chordates. Here it goes, about to roll. Going to see it here in just about a minute. There you go. There's the neural tube right there, zipping up. And now you see that the embryo is rolling. It's important for rolling that it ends up with this left-right asymmetry. And now the notochord is stacked up in what we call a stack of cells. All Ascidian embryos have 40 notochord cells. And what's going to happen now is that tail is going to wrap around the head. This also shows you one of the beauties of working with marine embryos is they're the same size. The larva turns out the same size as the egg. You can see the beautiful process of developmental biology, how you go from a single cell to a multicellular organism. You can see now that this tail is swelling. So what happens is you have the 40 notochord cells and then they swell and the tail is going to go up and around. You can kind of see right now, you can see those 40 notochord cells stacked up like a stack of coin. And this is really important, of course, for making the tail for the tadpole that's going to start to hatch here pretty soon. You'll see the test cells start to get really crazy. Try not to look at them. They're not the stars of this show, I'm telling you. It's the larva inside of there. But you can see them start going kind of crazy there. I should also say that this movie was taken with the help of students in some embryology courses that we give at Friday Harbor Labs. And now the tail is going to start twitching, so the embryo's getting ready to swim away here pretty soon. You can see the tail's wrapped all the way around. And the muscle cells, there's 3 muscle cells on either side of the notochord that are now causing it to twitch. And pretty soon you'll see it bust out of there and swim away. You can see that chorion's looking a little busted up here after sitting here under the microscope for all this time. There go the other cells. And it's ready to go free here. And we're really interested in what happens when the Ascidian tadpole loses this tail that it has, becomes completely tailless, plops out, metamorphoses into an adult, and it has a very, very different, completely different lifestyle then. You'll see it, here it goes. Ta-da! And then they're like, what? What happened here? Don't look at them. Don't look at those cells. I'm just kidding. This is some work done in Bill Smith's lab showing that you start out here. Let's see if I can do this without, oops, no, I can't. If you look in A, you can see the 10 cells that starts with the notochord cells, and then those go through 2 divisions, so B to a 20 cells, and then C to 40 cells. And then what they do is they stack up, they make this little pizza pie, as Ed Monroe called it, and then they go through convergent extension. And you can see in F, then they're all stacked up there, the 40 notochord cells. And so what we have found, and I'll show you, is that in the tailless species, every tailless species we've looked at has the notochord. And surprisingly, they have all the notochord genes, they have the notochord network intact, and they just make this what we call a nodal ball. So they're just stuck there with the nodal ball. In the hybrid, what happens is they go through convergent extension, and we're really trying to figure out what's going on in the hybrid that they're able to converge and extend that they don't have that gives them that nodal ball. And I'm going to show you that the other thing we're surprised about is they actually have a pretty good nervous system. I thought for sure the nervous system would just be gone on these things, but it turns out that it's not. So we know a lot about the notochord gene network through work Mike Levine and Anna Di Gregorio, and starts out with beta-catenin, and you can see just a small network there, but brachyury is a really important gene there. So we thought, brachyury, that's got to be it, you know, and so then we looked at brachyury, and you see in A and D is the tailed species, B and E is the tailless, or hybrids, and then the C and F is the tailless species, and you can see that actually brachyury is there at the right place at the right time, and seems to be intact and seems to be expressed just fine, which we were surprised about. Then we looked at some of these brachyury downstream genes. It turns out that those are all there and on in the right place at the right time in the nodal ball. So we still don't know what's not allowing them to converge and extend. But if you look, this is a confocal there, and you can see the notochord there in purple, pseudo-colored purple, and then you can see the neural tube above that then. So it might be that the notochord needs to be there because you have to make nerves, because you have to have the nervous system of the adult. So this is some work that we did in collaboration with Lionel Christian and Claudia Racicopi, who was at the Station Zoologica in Naples and then did a postdoc with Lionel and is now working in New York. And so what we were interested in was this black pigment spot. Again, that's made by tyrosinase and tyrosinase-associated proteins. It's very clearly there in the hybrids and not there in the tailless species. This is just showing that we cloned the right genes. Claudia cloned the right genes. And this is showing what we see when we look at these. And what we see is that let's see if I can use this instead. What you see is that there's these insertions or deletions of 5 to 7 base pairs. And so what happens is that completely blows the amino acid sequence apart. And these are pseudogenes. And what we found in the transcriptome is that a lot of these larval genes that we see are pseudogenes. So we think it's at an evolutionary time when eventually they're going to shut off. That's what molecular evolutionists will tell you. They're shut off if they're, once they become pseudogenes. But we can find these mutations and we can look at the gene and then realize that they're still being expressed. And so that would be non-functional even though we see it expressed. And so now we know in the hybrids, when we look at the transcriptomes, it's the tailed gene that's being expressed in these hybrids that's giving it that pigment spot. And so Alberto and Nadine really helped us with some of the imaging of these, and Alberto is interested in nerves, and so we decided to look at the nervous system. In Siona, the connectome has been looked at. There's 330 neurons in the larval brain, 25 cell types, and mostly simple unbraided shapes. Each cell has on average 49 synapses with other cells, and so we thought, well, I was like, there's not going to be any motor neurons in these guys, but it turns out this is kind of a summary here, and you can see that actually these tailless species don't know they're tailless, I guess, because they've got these papillae here, and they've got motor neurons, and most of the things that you would expect the central nervous system to have. And it might be because they need their nerves to become a functional adult, so they can't have those become pseudogenes. And we're not sure exactly what's going on there. We're still looking at it. But this is some work that, again, we did in collaboration with Alberto and Lionel. And you can see this is the nervous system here. They have these neuropores. And you can see there's some ciliated cells there, not as many as you find in the tailed species normally. And this is showing some of those. These are the motor neurons here, and you can see that if you label these, you can see that they're there, except for islet. Islet doesn't look like it's there, motor neuron. Islet's also in the notochord, and we think that's what that is there, is the nodal ball. So we do see some changes in the nervous system, and we're trying to figure out now exactly, what was that first hit that then is causing them to go, to shreds like that. One of the other things we're looking at in the lab is the Hox genes. It turns out we've heard a lot about the Hox, and you're looking at something like a lancelet here, Amphioxus, and then vertebrates and Saccoglossus, which even has a pretty intact Hox cluster. Ascidian's always kind of the weird one out. Here you can see that they've completely lost their middle genes. And that's the same whether they're colonial or solitary, and so we're trying to figure out now if that's has something to do with, they seem to not really follow collinearity either, and so we're trying to figure out now whether how those are looking between the tailed and tailless species, and if that's something that's causing what we're seeing as well. So to summarize then, I didn't show you all this, but I did this work with Titus Brown, and when he did the reassembly, he found this digital normalization that he uses a lot now. Both tailed and tailless species express the majority of known notochord genes, and they seem to be intact, whereas some of the other genes are pseudogenes. They have this nodal ball, but they don't converge and extend, and we really would like to see what's going on in the hybrids to have convergent extension. And many of those larval genes, mucilactin, tyrosinase, et cetera, are pseudogenes but still expressed. And I didn't show you this, but constructs can be electroporated, and now we're continuing to go back and look at gene function. So just to thank my collaborators, Titus Brown and Kanjan, and Titus was at MSU, he's now at UC Davis. Biennial was at NYU, is now at SARS, and Alberto and Elijah at Georgia Tech, and Elijah now has a job with the CDC, and Alberto is continuing to work on this. And then people in my lab that have worked on this, including Carrie Weber, who's here, and looked at a lot of these metamorphosis genes, and also Jennifer Ortiz and Megan Powers, who also are here and helped work at the thing. And so I still have a little time left. I'm proud of myself, and thank you very much. I'm very happy to be here, and there seems like we have so many friends and so much great science going on. It's been wonderful, invigorating, and I'm really, really thankful, and I really love evolution development and also developmental biology. So thank you very much.